Hello, everyone. Thank you so much for joining us today for this very special live stream. So excited to take a special look at how our restoration teams are working to better understand and restore deep sea corals in the Gulf. My name is Erin Spencer, and I am the communications manager for the mesophotic and deep benthic communities portfolio. And I will be your host today from Shoreside. And we have some really wonderful team members that are currently at sea right now on a vessel. And we're going to have an opportunity to connect with them in just a few minutes. But first, we're going to go over a little bit of background. They're currently on a ship called the RV Point Sur. It's a 135-foot vessel owned by the University of Southern Mississippi and operated by the Louisiana University's Marine Consortium, also known as LUMCON. Today, the team is located in the Gulf, approximately 60 miles south from Louisiana. And we'll have to ask them how the conditions are out there right now and see if they've got smooth sailing. Throughout today's program, please share any questions or comments that you have in the chat. We will do our best to get to all of those, and you can ask your questions in real time of the scientists. And just in a few minutes, we'll connect with those folks live. But first, we're going to talk a little bit about the background of MDBC. As you travel deeper in the ocean, less and less sunlight is able to penetrate the water. Just past recreational scuba diving limits, you reach something called the mesophotic zone, where mesophotic means middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, a variety of fish, and all kinds of invertebrates. Beyond this twilight zone, no sunlight reaches at all, and it's also much colder, and the pressure is much higher. But there's still vibrant habitats in the deep sea, full of life, even hundreds to thousands of feet below the surface. These mesophotic and deep benthic, or seafloor habitats, are found worldwide, including in the Gulf. However, the 2010 Deepwater Horizon oil spill dealt a heavy blow to Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted. More than 770 square miles of deep sea habitat were impacted by the spill. But as a result, collaborative restoration projects are underway to help us understand and restore these seafloor ecosystems. These projects are developing new and improved maps of the seafloor to locate these important coral communities, learning more about the species that live in these habitats, the environmental conditions they thrive in, and how they might be connected across the Gulf. Determining the best methods to encourage deep sea coral growth and reproduction to help restore impacted coral species and the habitats that they create. And lastly, reducing the threats that these ecosystems face, like marine debris, and connecting these habitats and restoration work to the public, like on these live streams, and natural resource managers. This restoration work is made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees, and is led by the National Oceanic and Atmospheric Administration and the Department of Interior. Plus, there's many partners, including a large number of academic institutions and non-governmental organizations. So we have a lot of folks on our team. You can also scan that QR code to learn more, but we'll also be sharing some links in the chat for where you can learn more about this work. Now, I promise we are going to get to our colleagues on the ship, but before we do, I want to also share this cool video as a background to kind of introduce some of the topics that we're going to be talking about today. All the way back in 2011, this was filmed with one of our colleagues, Dr. Eric Cordes, and he's going to share a little bit more about some of the specific corals that we're looking at on this mission.
We named the study Lophelia 2 mostly because it was focused around probably the poster child for deepwater corals, Lophelia pertusa. This is a species that has a worldwide distribution. It's the one that we really know the most about going into the study. We're trying to learn a lot more about its biology and its distribution in the Gulf of Mexico, but we're really starting to tease apart what makes it tick, why it shows up in the places where it's found, how it can form these large reef structures, and what kind of other organisms are attracted to the habitat that it makes in the deep sea. Lophelia is really well adapted to the deep water environment. It doesn't contain the photosynthetic algae as symbionts that a lot of the shallow water corals do. It tends to grow very slowly because it's entirely reliant on feeding from particles that rain down from the surface. And Lophelia is well adapted to these deep water conditions where there's no light, very low temperatures, and a lot of other oceanographic variables that we're really just beginning to understand.
Well, one of the great things is not only learning more about these species, but then also getting to share this wild, cool, deep water footage that you just got to see featured in this video. And hopefully we'll have a little bit more of that to share throughout the program. But at long last, let's go over to the ship and meet our scientists.
Okay.
Hi, everyone.
Hi.
Hey, please introduce yourselves and let us know what you're doing on board.
Hi, I'm Chris Tancredi. I am a PhD student in Dr. Cortez's lab, and I am looking at if there have been changes over time in what the Lophelia or Desmophyllum pertusum looks like in the Gulf. I'm also going to be doing some in-lab experiments to try to better understand the species and understand what it likes.
Super. So, hi, I'm Dr. Chris Kellogg with the U.S. Geological Survey, and I'm out here doing some coral microbiology.
And I'm Morgan Will. I'm also a PhD student in the Cortez lab at Temple University, and I study more of the communities that live with the Desmophyllum pertusum, so all of the urchins and sea stars and everything that live on them.
Awesome. So one thing that I definitely want to start off with, because we just saw in the video with Dr. Cortez, him talking about Lophelia, but then Chris, you mentioned Lophelia / Desmophyllum pertusum. So this is actually a really interesting part of this species story. Could someone tell us a little bit more about why that is?
Sure, I will take a stab at it. So let me see if I can show you. So here's a piece, get it closer to the camera, of thanks, Chris, of Desmophyllum pertusum. And you can see each of these little nodes where it sort of has elbows. Each one of those is a place that a polyp sits. So it's a colonial coral with multiple animals on a single branch. And at some point, someone was looking at that and also looked at a coral that's a single polyp cup coral named Desmophyllum dianthus and said, you know, these kind of have some characteristics in common. We should maybe look at the genetics. And so they effectively did sort of an ancestry.com comparison to look at this coral and that coral and see how closely related they were. And we're kind of surprised because people thought, well, maybe they're cousins, and their genetics suggested that they were more like siblings. And so because of that, they changed the name of this coral to be Desmophyllum pertusum, so it would be in the same genus as Desmophyllum dianthus. Now, having said that, people may have heard of Stephanie Germanata, but most people know her by her more popular stage name of Lady Gaga. And so similarly, while this is the taxonomically accepted government name, Desmophyllum pertusum, a lot of people, including some of us who have worked on it for 20 plus years, will still sometimes slip and refer to it by its much more popular stage name of Lophelia. So 2 names, same coral, this one.
What an amazing comparison. And now I won't be able to look at the coral without thinking of Lady Gaga, which I love. You know, and this is something that comes up quite a lot, right? In science, we see that as we learn more information about species, that we actually, things can change. What we thought we knew before isn't the case. Is this something that we're seeing quite frequently in deep sea work?
I think it is, just because a lot of times, well, it's unusual, I think, to see something change because there's so much that we don't know. So I think in most deep sea cases, we're just at the first name or the first time we've seen it and giving it a brand new name, as opposed to something like this where people have studied Desmophyllum pertusum for a long time. I'd say it was sort of the poster child of deep sea corals because it creates these, very big, visually attractive mounds like the video you were showing. And so because there's been a lot of research focused on that particular species, we knew enough to then start iterating on what we know about it. Whereas much of the other work, the first time we see something might be the first time. And so a lot of it is very new.
And so going more specifically into Desmophyllum pertusum, what makes this species a little bit different than maybe some of the other mesophotic and deep coral species that we see in the Gulf?
I can take that one. So a lot of the corals that we see in the deep sea are big fans that we call soft corals. And that's going to be like what you're seeing behind Erin in her little screensaver. It's a big fan. Whereas Lophelia is what that's a beautiful fan. Whereas Lophelia is what we call a hard coral. So it forms these really hard calcareous skeletons that give it a lot of extra structure. It allows it to grow a lot bigger and provide a lot of support for other creatures to live on. So it provides a ton of habitat and creates these really cool communities that are all related to it.
Awesome. And some gorgeous, gorgeous footage to go along with that. Such a cool comparison to see those different species. And thinking specifically about some of the goals of this particular expedition, I know that some of the sites that you are visiting now are actually sites that we've been to before, both in the years before and immediately after the Deepwater Horizon oil spill. How will the data and images that you're collecting now help us better understand these sites, specifically how it'll help us understand how they have recovered since Deepwater Horizon?
Take that, Chris.
Yeah, I can take that. So the work that I'm specifically doing is looking at video over time. So I'm looking at video that was collected in 2009 before the oil spill, 2010, 2013, 2014, and so on. And when I look at this video, we want to make sure that we're going over some of the same places over and over again so that we can see how the coral health has progressed. And we want to be able to determine not only if there have been decreases, but also if there's been evidence of resilience or recovery. So some of what we're doing is establishing a baseline, and we can look at that baseline for later to see how the coral community has recovered.
Yeah, you can see on screen right now one of the markers, and that's something we were doing yesterday at a site, was going from place to place and looking for some of those same markers so we could line up and make sure we were in exactly the place and the orientation that had been previously visited so we could get good photos and video and do those kind of comparisons.
Now, is it hard to find those markers again? It feels like it would be searching for a needle in a haystack.
It's interestingly difficult and not difficult at the same time. So we have lats and longs for them that we can target and go for. And sometimes we land right on them, and sometimes we drive in circles and don't see them at all. And other times we're looking for one and we find someone that we weren't looking for at all. So it's very interesting. As you get down deeper, it's harder to get exact navigation. The lat and long gets less and less exact the deeper you go. So at the sites we've been at in this cruise, it's been pretty reasonable to find them at these depths.
That's great.
Yeah, going along with that, we were able to find some of the ones that I specifically was looking for. And it's helpful because I think people in the past have had this great idea to make them a little bit reflective. So we are able to see them a little bit easier than if you're just like looking for something that's not reflective at all. When we shine a light on it, sometimes they're like, oh my gosh, there's a marker there, and we're able to find it a little bit better. But it is sometimes difficult. The markers aren't always where you think they are, or may have had something happen.
Or are covered in anemones.
Yes, we see that a lot. There's covered in things, and you can't even tell what they say anymore. So, yeah.
The unexpected parts of fieldwork, right? When anemones get in the way of finding your spot. Now, you all are talking about how you're finding these. I want to switch here. This is actually I think you're doing some ROV footage as we speak. So this is the live ROV footage. So can you describe, you all are standing in the lab right now. Where are the ROV operations happening on board?
Sure. So actually sort of right over Chris's shoulder, there's a bank, and the monitors, the ROV pilot, and the person that would handle the manipulator arm to take samples are sitting right over there. So in general, we're all in one room together. So they're sort of right behind us.
Oh my gosh,
Perfect.
This is such a great mobile cam view happening just behind. Yeah, we can see you guys all the way in the back. I think it's also interesting to note that I think some people don't understand how many things can be happening at the same time while you're on board. In this case, very specifically, you guys are operating a live stream at the same time the ROV is being deployed. But can you tell us a little bit more about what your day in the lab while you're on board?
Sure, I'll start. So we get up in the morning. There's a whiteboard where the plant of the day usually lives. And so typically first they'll take a CTD cast, so conductivity, depth, and temperature, where they put bottles on a carousel over the side, and we get water samples. And that gives us a sense of temperature and salinity for the site. Also, some of those samples get filtered to look at nutrients or particulate organic matter, or sometimes they just need to collect bottom water that's cold for when we have the live corals, because those get water changed. And so typically then the water changes happen because the corals get fresh water every day. So the ones that we have collected that are going to be brought back are living at a spa. And then the first ROV goes in and we start doing what you saw. We all watch the screens and try and pick out what we're seeing. And if we see a target species that we were maybe wanting to sample, then that will also happen. When the ROV comes back, we all run out there and bring the samples back in, and then things, really get crazy. So I'm usually doing a little bit of preserving samples to do microbiology back in the lab. The folks from the Smithsonian Natural History Museum are going through and looking at things and subsampling those for reference vouchers and other work. I know, trying to think what else is, some of each of the different samples are being processed a tiny bit for stable isotopes, which goes into figuring out food webs, who's eating who and what are they eating. So lots of things. So long periods of us just watching screens, and then burst of activity when the ROV comes back up with samples.
And I think it also is important to know just how many types of data you're getting with each expedition that you're on. And that leads me to something that I want to go back to, which is actually for Morgan and Chris. You both mentioned that you are graduate students. Can you tell us a little bit more about what you're hoping to get out of your work, and also how that applies to restoration? And meanwhile, we can enjoy this gorgeous ROV footage as it's going.
Sure, I can go. So I am mainly interested in communities. So a lot of my work is actually watching the footage that you're watching right now. I will watch this when we get back to shore, and I will ID and count all of the things you're seeing. And then I take all that data and run some wonderful stats so that we can basically compare the communities we're seeing at this site to other sites, or the communities we're seeing at this site now versus the past, or setting it up for, are we going to see changes, are we going to see changes in the future? So I love the communities. I look at kind of all the critters that live with the corals is my main focus, and which relates really well to this work, because whereas MDBC is mostly focused on the corals provide habitat to everything else. That's why we really care about them. So looking at how all the communities around them are changing really helps us understand if our restoration efforts are making a big impact to the ecosystem as a whole. You want to go?
And hi again, I'm Chris, and I'm looking at whether or not there have been shifts from, like, at the more than just the colony level, but within the sites that we're looking at, from this live coral, which we were showing earlier, that looks kind of like this, it's very white, to something that looks more like this. So it's brown, and it actually gets coated, I believe, in manganese, and it basically the structure of the coral is still there. It still provides this habitat when it's dead, but it might be more susceptible to things like breakage, especially as ocean acidification is occurring. So I'm trying to determine if this shift, if there's a shift that's occurring from these communities, or this population that's very alive, to a population that's more dead. And there has been some evidence maybe that different communities might live within the different types of structural habitats. So areas that are more alive might contain different critters than areas that are more dead. So this kind of goes along with things that other people are working on. And so I'm just trying to determine if there have been changes over time, and then using our knowledge about if there have been changes over time to inform kind of some of our next steps. And the other thing that I'm doing is we are collecting some live specimens. So I'm collecting corals that we're putting right now into quivers, which are like these tubes. And we bring them up, and then we store them, and we give them water changes. Oh, that's a bio box.
Mm-hmm.
And so we're able to sample things, and I'm going to be trying to determine what the coral's like.
Amazing. It sounds like both of you are tackling some super important questions and also doing quite a bit as part of your dissertation research. And you mentioned looking at samples, and I wonder, is there an opportunity for us to see some of the samples that are currently on board?
Yes, I think so. Yes. We should be able to do that.
Mm-hmm.
Could I have somebody help me open this port? Wonderful. Okay,
amazing. We've got our mobile cam on the go.
Okay, can you put this back down? I'm going to rest it right on top. Okay, so here's an example. That one actually has its polyps extended, so this one's doing pretty well. Someone's really happy in here. So when they're looking very healthy, you can see that they have little tentacles right here that are extended. And yeah, so this is an example of one of ours that's doing particularly well. And I'm going to bring this one hopefully back to the lab.
That's awesome.
And very alive.
What's involved in keeping, you said this particular sample looks really happy. What's involved in husbandry on board?
Honestly, these are pretty hardy. So what we're doing is we're making sure that their water remains relatively clean, and we're keeping them cold. So they're in this giant chest freezer that we have pulled them out of. And so this is kept at around 8 degrees Celsius. And so we make sure that their water is kept cold. And every day we open up these containers and we remove some of the water and add fresh water that's been filtered.
Okay, and that goes back to what Chris was talking about, was when you, like the CTD cast in the morning, is that right, as part of where you're getting samples or getting clean water for the corals?
Yes, that is, and the water ends up getting filtered. And then we get to use that.
Okay, well, thank you so much for showing us the live corals. This is so exciting to think about the corals coming from the seafloor, kept happy and healthy on board, and then will contribute to all sorts of great questions that we have about restoration. And so don't forget to, if you have any questions, make sure to put them in the chat. Otherwise, I'll just keep asking my own questions, but I want to make sure everyone has an opportunity. While folks are thinking of their questions, I will ask another, which is, so far on this expedition, what is one of the most interesting or unexpected things that you've seen on the ROV footage?
Ooh, well, I'm not sure it's unexpected. It was really cool for me. It was the first time I've seen it. Yesterday, we saw this huge tube worm bush conglomeration, and it's been studied for a long time, and there were a bunch of markers in it, one of which was a really big lander in the middle, which was somewhat difficult to find, but we basically flew over these huge tube worm bushes for a while until we found this big lander that was covered in Calogorgia, which is a type of fan. So there's this coral, this little island of coral floating in the middle of this giant tube worm bush field. So I think we were expecting that. Someone put the lander there before, the float, but it was really cool for me. It's the first time I've seen anything like that. Does anyone else have
Again, so I study microbes, so I don't ever expect to come out here and see the things I'm studying until I get back to the lab. But got to admit, I live for the cephalopods, and so far we've seen a couple of octopus and a bobtail squid, and then there was definitely a big red squid today. So I'm always glued to the feed watching for one of those because they tend to go really fast.
I really like seeing the area in a way that I can more easily interact with it and tell like what exactly is going on. I think it's really fun to have like the nav the navigation data right in front of you. You can see where the ROV is, you can see where the ship is, and you can see where you think you are on your site. And it's really nice for me to be able to see the front end of kind of what I've been doing research and work on in the back end.
That all sounds amazing. And I especially love the idea of these like huge mounds of tube worm fields. That sounds like it was a great site. We do actually have a question in the chat that I want to toss from Dave. Thinking back to the sample that you showed, he's asking, are the tentacles for eating?
Yeah, they do use them to eat, yeah. They filter feed, so they have their tentacles in the water, and they rely on, as Dr. Cordes said in the video, this thing called particulate organic carbon or particulate organic matter or marine snow, and it kind of drifts down, and the currents bring it the food that it needs, and it kind of grab it with its tentacles.
Because a reminder that because these corals are living in total darkness, other than when we come up and shine a light on them from the ROV, they're not like tropical corals that you would see when you're snorkeling that have photosynthetic algae in them. And so, yes, they don't have something inside that's manufacturing their food. They've got to get out there and do the work themselves.
And we have some wonderful footage that was just showing in that small scale of looking at those little coral tentacles pulling in their food. But also a great reminder, you know, a lot of us are most familiar with those shallow water corals, like in the Florida Keys and in the shallower parts of Flower Garden Banks National Marine Sanctuary. And of course, they have all that delicious sunlight coming in that the deepwater corals can't rely on. Okay. We have a couple more minutes if you want to add your questions in the chat. I see someone was asking about an invertebrate ID guide. There are a couple of links in the chat for where you can find some existing ID guides. And so I will toss the question back to you all on board. What is one of the things, because, you know, of course, you're very busy on board and you're in the nitty gritty and thinking about the data, but it's taking a step back into the big picture. What are some of the things that make you the most excited and hopeful about this sort of work? Because, you know, we're getting there's a lot of tough news out there when it comes to the ocean and the threats facing our ocean. And so I think but this is a really interesting portfolio. It's a great opportunity to talk about hope.
Sure. For me, and it's, you know, this cruise and doing this kind of field work in general, I really look forward to it because for me, I take these samples back to the lab and there's so much unknown about the deep sea, even now as we're coming out, like you said, to sites we visited before. You're still pretty much guaranteed to learn something new. And for me, that's why I love my job, is being in the lab, working on something, and then you discover something new. So, you know, I might identify a new microbe or an unexpected association. And there's, for a few seconds, when you know something that you've just discovered that nobody else knows, is amazing. And then even more amazing is when you then run down the hall and start telling everybody else about it, because it's this new piece of information that you've just discovered. And it's just an amazing feeling to find new information and know, not just for yourself, which is exciting, but to then add that to this, you know, large history that we have of scientific knowledge, and know that you've added something new and built on that then other people, students like these two, will then take and keep going. And so that knowledge keeps moving forward. And I love that.
Amazing.
And I mean, as far as what makes just seeing these communities, they're all gorgeous. So you can hear a lot in the news about how, you know, a lot of stuff is dying and there's all kinds of issues, but these communities, even if they are affected, even the effects that they're having, they're still thriving in their own way. They're resilient, they're coming back. So being on these cruises, you actually really get to see, we were at Viascanol a few days ago, and even though we're a little worried about some of it, it's still gorgeous. And there's still so many creatures there. So that does give you a little bit of hope still looking at it. That even if it's a little different, it's still, Mother Nature moves on, right? So we're still tracking forward.
I would like to second that same thought, because the site that I've been working at, I'm looking at, and I'm trying to figure out kind of what's been happening. And we came upon this marker that we weren't expecting to find. And there was just this giant, beautiful mound of coral there, and it was amazing to see. I think everybody kind of just like had this collective like when we saw it, and it was really an amazing thing to see. So I enjoy the discovery part of it. We're learning new things, and we're finding old things. So I think that's my favorite part.
And then I think a great way to end is thinking about how your work specifically, and then also what you're doing together as part of this team on the expedition, contributes to the restoration of these deep sea and mesophotic habitats. So could you touch on that as well?
Sure. Yeah. So there are a lot of things that are being done for restoration beyond just what's being done by the people in this room. There have been several projects that have been worked on, but we really, in order to determine what we need to do next, need to understand what has happened. So it's not that we are only doing like damage assessment, but we need to be able to determine what the current situation is for these corals so that we can move forward. So we're still in some early work with this. There's a lot more being done, but yeah, that, and there's also people are doing coral propagation. People are trying to determine what the corals like, what is best suited for them to be kept in the lab. I think there are also some potential transplantation efforts. I'm not sure if there's any with Lophelia, but there are a bunch of different things being done for trying to restore this coral.
There's the lander. I'm sorry, that's what I was talking about.
Yes.
Caligorgia. Sorry.
Yes.
Okay. Had to put it out.
And this is actually a great tie-in. Can you tell us a little bit about the lander and kind of how that also ties into the restoration? I know that's not your exact area of research, but since you brought it up.
Sorry, I mean, this ties more into, like, we're trying to keep track of what these communities look like over time. Really know. So this one we've obviously been to before. That's why this is here. So we're going to it now to see what it looks like now, so that we can again go back in the future and keep a good time track. So as we're recovering from this big oil spill, we're watching the restoration in progress that's happening both naturally, which is what we're watching here, as well as our efforts in more of the lab setting to try and aid in that restoration. So having these big markers out is just a really good way of keeping really good track of all of our increments as the restoration moves forward.
Awesome. And then any last thoughts before we wrap up? I would just want to say thank you all so much for taking time out of the busy work that's on board to chat with us. It's really been wonderful to hear your perspectives and the sort of restoration work that you're doing and how that ties to this bigger body of work. So I would say if you have any other last thoughts, I'll check for any other questions in the chat. One comment says to say hi to Chris.
For me.
Both, maybe both Chris's. We always love it too when folks are able to tune in and from land and see folks that they know. So, okay, I think that is a great way to wrap up. Again, thank you all so much for your time. Love to see this ROV footage and see the live samples and see all of this wonderful stuff that you're doing. So please remember that you can continue to tune into these live streams. We have a couple of others that are coming up over the next few weeks. If you like today's event, tell your network. We're going to put a link in the chat to show where you can find our upcoming live streams. You can also watch recordings of past live streams and see other upcoming events on the University of Rhode Island Inner Space Center's YouTube page. To learn more about these projects, find articles, publications, and our story map, all kinds of things, check out the links that are going to be pasted in the chat. And then one last thing is we would love to get your feedback on today's program. So if you wouldn't mind filling out a quick survey that will also be in the chat, we would greatly appreciate that. So with that, I'll say thank you again to our wonderful speakers. Thank you again to everyone who tuned in online and in the future in these recorded live streams. And don't forget to join us again. But we really appreciate it.